Hello, my name is Corey James, and the article that I'm going to be presenting is titled Gut Bacteria Driven Homovanillic Acid Alleviates Depression by Modulating Synaptic Integrity. And this was in the Cell Metabolism Journal in May of 2024. So, depression is a major health burden worldwide, with around 300 million people in the world having been affected by major depressive disorder, or MDD, with pregnant women, the elderly, and children ranking among those experiencing the highest incidence of MDD. Symptoms of MDD include low mood, loss of interest in life, fatigue, delayed thinking, and repeated fluctuations in mental state. Depression is complex and it involves genetic and environmental factors, and the pathology of this disease is not well understood. One theory of the pathology is the neurotransmitter and receptor hypothesis, which suggests certain neurotransmitter deficiencies are at the root of the cause of MDD. Current clinical medications often have limited effectiveness and may actually exacerbate symptoms, leading to a strong need for new therapies and medication options. The gut brain axis, or I added microbiota gut brain axis, is a bidirectional communication between the gut and the brain, including communication between gut microbiota and metabolites, and has become a recent target for possible treatments for depression. Thus, understanding the relationship between the gut and depression is necessary. Most previous literature discusses gut microbes as diagnostic markers for depression. More recent research establishes a causal relationship between depression severity and high serum proline levels, where proline levels were associated with a unique microbiome signature, as well as dietary traits, opening the door to the idea that metabolites not only play a role in depression, but are possibly being a causal agent in the severity of depression symptoms. So, the highlights from this study, or the bluff, include that HVA levels are depleted along with altered gut bacteria in people with depression. The bacteria R. intestinalis promotes the abundance of B. longum for HVA production. Treatment with HVA, B. longum, or R. intestinalis alleviates depression in mice. And HVA restores synaptic function by inhibiting autophagic death. So, what is HVA? Since I mentioned it in almost every one of those bluffs. HVA stands for homovanillic acid, which is a byproduct of the catabolism of dopamine. I think we've all heard of dopamine. It's an important chemical that affects mood, motivation, and attention. So, HVA levels can be measured to get a sense of the function of dopamine in our bodies. HVA levels in urine have also been used to diagnose certain types of tumors and other metabolic disorders. And HVA levels in the brain have also been associated with schizophrenia. The overall goal of this study was to elucidate complex interactions and molecular mechanisms underpinning the gut microbiota brain axis in depression to pave new avenues for the development of targeted therapies in the management of depression. So there's a couple of different methods and studies that they did here. One of them involves the human study. So I want to spend a little bit of time explaining the setup, and hopefully that'll make the results easier to understand later. So for our human study, they collected serum for metabolic profiling and fecal samples for the metagenomic analysis from the First Hospital of Shaanxi Medical University and Shanghai Mental Health Center. They did obtain informed consent, as well as approval by the ethics committee of the hospital and mental health center to be in compliance with the World Medical Association's Declaration of Helsinki. There are two human cohorts where samples were collected from controls and individuals with depression. And those with depression had been diagnosed based on the HAM-D17 scores. HAM-D17 is the Hamilton Depression Rating Scale, specifically the 17-item version. And the scoring suggested by Zimmerman goes as follows. So I have those listed here. No depression would be a 0 through 7, mild depression 8 through 16, moderate depression 17 to 23, and severe depression is greater than or equal to 24. We see that there are 91 controls and 155 people with depression in the first cohort, and we have 116 controls and 107 people with depression in the second cohort. And then I wanted just to have you note the median HAM-D17 scores. We have 8.15 for the controls and 22.53 for the depressed group, and 3.79 versus 21.41 for cohort 2. They didn't indicate what cutoff was used for diagnosing depression, so I couldn't tell you at what level they were determined to have depression for this particular study. And no one in either cohort is taking medication. Okay, this gets a little more complicated. So all animal studies were approved by the Institutional Animal Care and Use Committee at the Center for Laboratory Animals. They used normal mice and germ-free mice, which were acclimated in an animal facility with chow diet and ad libitum for 1 week. The Combs and Court mouse models are used to induce a depression phenotype in mice and are commonly used in the study of depression. Here they had 5 different animal experiments. So in experiment 1, the Combs model test, or chronic unpredictable mild stress. Combs mice were divided into 2 groups, the control group and Combs group. The Combs group exposed to unpredictable stressors daily. They could have had 5 hours in a cold room. They could have had a soiled cage, water deprivation, reverse the day and night. They could have had restraints, electric stimulation, or fasting. And after the weeks that they did that, they did behavior testing on both groups at week 6. And the behavior testing included a forced swim test, tail suspension test, and sucrose preference test. In experiment 2, it's the HVA homovanillic acid intervention in Combs model. So kind of repeat the same setup, exposing the Combs mouse group to the same stressors that was in experiment 1. And then you divide that group into 3 groups. You have one that's just Combs, but then you have 2 with interventions, Combs plus HVA at 100 milligrams per kilogram, and one that was Combs plus HVA with HVA at 300 milligrams per kilogram. And then they followed with the behavior testing again. In experiment 3, we have HVA B longum or R intestinalis intervention in the Combs model. So again, mice, you have the Combs mice, and they were divided into 4 groups, Combs plus HVA, and that was at the 300 milligrams per kilogram, Combs plus B longum, and Combs plus R intestinalis, followed by behavior testing. Experiment 4 is the Court model, Court being corticosterone-induced depression. There was 2 groups. There was a control and a Court group, which was injected subcutaneously with corticosterone for 5 weeks, and then followed by the behavior experiments. And then in experiment 5, you have HVA, B longum, or R intestinalis intervention in the Court model. So you have your control group and Court with the interventions. So Court plus HVA, Court plus B longum, or Court plus R intestinalis. And at the end of the behavior test, you had behavior testing again. And then after all of these experiments, all mice were euthanized and the serum, intestinal, and brain specimens were collected. All right, total microbial genomic DNA was extracted from fecal samples from the human cohort and cecal contents from the mouse model experiments using the DNeasy PowerSoil toolkit. Nanodrop Indy 1000 spectrophotometer and gel electrophoresis were used to measure the DNA concentration, purity, and quality. A metagenomic shotgun library was constructed using Illumina, TrueSeq NanoDNA LT library prep kit. Paired in sequencing with 150 sequences per read was utilized. And then sequenced reads were compared to the host genome, so host reads could be removed. Quality reads were recombined to produce a macrogenome for each sample, and BLASTIN was used to determine the relative abundance distribution. Analysis of the relative abundance of B. longum followed a similar process, with the extracted microbial DNA being processed to construct cybergreen-based quantitative RT-PCR. The primers for B. longum and 16S are found in table S3 right here. Metabolic profiling was performed on serum samples from the human cohort and tissues of the mice using the Q300 metabolite assay kit. Liquid chromatography and mass spectrometry were performed. Bacterial strains were purchased. R. intestinalis, and R. fasciae were cultured using DSMZ mediums, while B. longum and E. rectale were cultured using ATCC mediums. All were cultured at 37 degrees under anaerobic conditions. Methods for the cell experiments included hippocampal neuron isolation of the mice, after various experiments were performed, followed by synaptic experiments on some of the neurons, and autophagic death or flux experiments on others. Western blot with brain tissue was also performed, as well as immunofluorescent staining and transmission electron microscope staining. Data is presented as a mean plus or minus SEM values, or standard error of the mean. Various statistical analyses were utilized, including Spearman correlation, principal component analysis, linear discriminant, effect size, partial least squares, logistic, and linear regressions. Okay, so starting with the human cohort, we had 207 controls, 262 individuals in the depression group. Blood samples were collected from all participants, and fecal samples were collected from a subset of the group. So only 129 fecal samples were collected. Here we see a close-up of panels 1B and C, as well as figure S1A from the supplemental materials. Panel B shows PLS-DA, which is partial least squares discriminant analysis scores, based on the metagenomic sequencing of fecal samples. We can see that there's a significant difference in the gut microbiome composition between controls and the depression group, controls being in blue and depression in pink. In panel C, we see that most of the differing bacteria belong to 4 phyla. They're listed up here: the Firmicutes, Proteobacteria, if I'm butchering these, I'm sorry, Proteobacteria, Bacteroidetes, and Actinobacteria. I included the supplementary figure. I know it's really tiny and we're not supposed to do that, but it shows the differences between the controls and depression group at the species level, and I just thought that was an interesting visual. So panel D shows the PLS-DA scores for the metabolite analysis, so right here, where a total of 203 metabolites were quantitated. You can see again that there are significant differences between the two groups. Controls are blue, the depression group is pink. And panel E is a volcano plot showing that neurotransmitters and bile salts are the metabolite classes with the biggest differences. So we have the neurotransmitters here, and, well, I said bile salt, bile acids. With the colors representing the effect size, pink is 0.2, ranging down to blue at a negative 0.4. And the size of the bubble represents the negative natural log of the p-value, which is often used for visualizing very small p-values. In this case, the larger the dot, the smaller the p-value. Supplemental 1B figure demonstrates that HVA was one of the neurotransmitters that differed significantly between the control and depression groups. We see HVA way over here. And then you have serotonin, L-tryptophan, and L-tyrosine as well. And then the authors in this study were focused on the neurotransmitters, but you can also see that bile acids GUDCA and CDCA were also significantly different here as well. So I just thought that was interesting to make note of. Here in panel 1F, we see that HVA, L-tryptophan, and L-tyrosine have the most pronounced effect sizes. You can see those here. So they were selected for further analysis. And in this case, all three are seen in a reduced abundance. Here, this figure 1G is a Sankey diagram showing the correlations between the three metabolites. So we have our metabolites up here. Bacteria and the Hamilton depression scores. So the HAM-D scores are down here. There is a significantly negative correlation between the three metabolites and the HAM-D score. So you can see those here. They're in kind of this bluish purple color, the negative associations. HVA has the most robust associations with bacteria, where you can see all those over here, both positive and negative, and it involves all but like these last two bacteria. HVA specifically had a positive correlation with Roseburia intestinalis, Bifidobacterium longum, Roseburia facies, and Eubacterium rectale. You can see that both here and in 1H. And the relative abundance of the four bacteria is correlated with HVA. So we have a correlation between the relative abundances, and that's interesting. However, something interesting to know is that the bacterial abundances were all decreased in the depression group compared to controls. So I think that's just something that's interesting to keep in mind. And then not shown is that HVA levels were also found to be lower in the depression groups in both serum and feces, which provides strong evidence that HVA plays a pivotal role in linking the gut microbiome and depression. So moving on to figure 2 and trying to determine if HVA is a gut microbiome derived metabolite, we see that panels B through D are showing the concentration of HVA in the gastrointestinal tract, the brain, and serum in germ-free mice compared to SPF or specific pathogen-free mice, with the concentrations of HVA being higher in all three for the SPF mice, indicating a significant contribution to HVA produced by the gut microbiome, because the germ-free mice don't have one. To determine if any of the four bacterial species previously determined to be related to HVA are producing HVA, the four bacteria were cultured individually. We see that here. In vitro, using mouse fodder as substrate, panel 2E shows that B. longum actually produces HVA while the others do not. And because the abundance of the four bacteria were all correlated with HVA, but no HVA was produced by three of the four in isolation, it was speculated that they may assist production of HVA in B. longum. Mice were administered one of the three bacteria, and R. intestinalis was found to be significantly increased in the abundance of B. longum. So you see that here. So looking at the concentration or the abundance of B. longum in the mice that were administered this R. intestinalis, we see an increased abundance in B. longum, but not so much with the others. And then also we see the same result in HVA. So the mice that were administered in R. intestinalis also had an increase in HVA, which indicates that R. intestinalis enhances the abundance of B. longum and in turn increase the production of HVA. Something to note is that this could not be replicated in vitro, which suggested to the authors that the unique environment of the gut microbiome is an important factor in this interplay. And I just wanted to remind you, we saw previously that B. longum abundance in those with depression is lower than those who do not have depression. To determine if the mouse models would replicate the reduced bacterial abundance and HVA concentrations seen in depressed humans, Samples were taken after the Combs and Court experiments. We see a significant reduction in both R. intestinalis and B. longum in both the Combs and Court models, as well as a reduced concentration of HVA in both the Combs and Court models, which is what we would expect if the mouse models follow what we already saw in the human cohorts. So we can see here that um, these bacteria were found in a lower abundance in the Combs model, or mice experiencing depressive like symptoms, same in the Court, and then also the reduction in HVA. So, investigating whether HVA or the gut microbes could have a therapeutic effect on depression, the Combs and Court mice were administered HVA, B. longum, or R. intestinalis twice a day for 5 weeks. The behavior tests were administered after the 5 weeks. We see a measure of immobility. So you can see these immobility measures. Immobility time, where immobility time is being used as an indicator of depressed phenotype in mice. A higher immobility time means more severe depression. So what we see here after, and I'm going to call them the interventions, but administering HVA or the bacteria, we see a significant reduction in immobility time in all cases compared to the Combs or Court without the intervention. So you see the red bar, that shows you what the Combs model looked like without any interventions. And then the yellow, green, and pink are all reduced in the immobility time for all of these. And then looking at the sucrose consumption, which is these last two, sucrose consumption tends to be higher under normal conditions. And so we see an increased sucrose consumption in panels H and I, nearly reaching the levels of the control. And bacterial and HVA interventions appear to improve depressed phenotypes in mice. That's the overall takeaway. Okay, so here the bacterial and HVA interventions also increased the expression of mature brain-derived neurotropic factor, or MBDNF. Previous research has indicated that MBDNF is significantly involved in the pathophysiology of major depressive disorder and could also play a role in the mechanisms of antidepressants. It has been reported that the levels of this protein are lower in patients with MDD, and there is a negative correlation between these serum levels and the severity of depression symptoms, meaning that lower MBDNF levels mean increased depression symptoms. So, seeing this increased protein expression is compelling and demonstrates a specific physiological change directly related to HVA levels and depression symptom severity. To determine if gut-derived HVA could cross the blood-brain barrier and exert antidepressant effects in the brain, isotope-labeled HVA was administered to mice. You can see in panel A that the HVA was detected in the medulla oblongata, hippocampus, and prefrontal cortex after oral administration. Panel B shows the HVA brain levels for HVA-treated Combs mice. And in C, you have increased HVA levels in both HVA and B. longum treated Court mice. This provides evidence that gut-derived HVA crosses the blood-brain barrier. Here we're looking at investigation of synaptic damage in the hippocampus, which is thought to be one of the main factors in the pathology of depression. Two proteins that can be used to study synaptic function are SIGN-1, which is involved in the control of neurotransmitter release at the presynaptic terminal, and PSD95, which is a postsynaptic scaffolding protein in excitatory neurons. This investigation into the plasticity of the synapses in the Combs and Court mouse models using immunofluorescence with HVA, B longum or R intestinalis interventions showed that sign 1 in the hippocampus is significantly upregulated in treated mice. So using the control as a reference for healthy synapse function, we're looking here, and understanding that sign 1 is dyed green, we see that in CUMS mice, sign 1 expression is severely reduced. And then as the interventions are performed, we see synaptic function returning as we look at each of these different interventions. It's returning more closely to what resembles the control coloration. There was not a significant difference involving PSD95, so that's this red panel, suggesting that the HVA intervention affects presynaptic dysfunction associated with depression, as opposed to the postsynaptic. Previous research has suggested there is excessive autophagy, I've heard this said a few ways, in synaptic damage, which in turn can contribute to the depression phenotype via the accumulation of degraded material that is not cleared out quickly enough. LC3 and SQSTM1/P62 are both proteins involved in the creation of the autophagosome and play an important role in efficient autophagy. This final analysis attempted to decipher how HVA might affect autophagy. LC3 and SQSTM1/P62 are both biochemical markers of autophagy in the dentate gyrus region of the hippocampus. Excessive autophagy was observed in the depressed mouse models with decreased expression of LC3 and SQSTM1, which you can see from like the controls to the CUMS and CORT models. And then with HVA intervention, expression levels returned to normal. This was also observed in the HVA plus B longum and R intestinalis groups. So further analysis determined that HVA exerts inhibitory effects on autophagy by managing the protein levels and limiting autophagosome quantities. So depression is a major issue worldwide and is a complex disease that involves genetics as well as a variety of environmental factors, and treatments for depression are lacking. It was previously known that there are alterations to the gut microbiota and metabolites in people with depression, and that there is a connection between the gut and brain function. Here it was found that HVA levels were reduced in people with depression, as well as certain gut bacteria. A metabolic association between the gut bacteria and HVA was identified, with the determination that B longum directly produces HVA, and B longum growth is enhanced by another gut bacteria, R intestinalis. Gut-produced HVA crosses the gut-brain axis and is effective at alleviating depressive phenotypes in mouse models. HVA exhibits neurotropic and neuroprotective effects via the prevention of sign 1 degradation and reducing autophagy in synapses. With this new understanding, the authors suggest that oral supplementation of HVA combined with a mixed probiotic cocktail could effectively manage depression symptoms. Broader implications include bringing us one step closer to understanding the connection between the gut microbiome and how it interacts with metabolites and other processes, specifically the pathology of depression and depression symptoms. Every new thing that we understand opens the door to novel ways to treat diseases, or at the very least, make current methods more effective. We still do not know if these findings will translate to a human model. Also, diet was not addressed in the human cohort for this study, which is a major factor in the gut microbiome. So exploring diet and how it plays a role is imperative. It was also discovered that certain bile acids were found in significantly varied levels, but this study did not explore how they might play a role in depression or disease severity. So further research should be done to elucidate how bile acids might also play a role in the pathology of depression. So really quickly, some of the limitations. Diet was not addressed, which could affect results, like I already mentioned. Also, the authors mentioned that bacteria capable of metabolizing HVA was not actually cultured from fecal samples of individuals with depression. So that's something that maybe would need to be done in the future. And then there also could be substrates other than tyrosine for HVA production. So another thing to explore. Future research could address how R. intestinalis regulates the growth of B. longum. I already mentioned bile acids were increased but not explored, so that would be something else to look at. We need to do more research to understand the underlying mechanisms in depression in the gut microbiome, and also maybe perform a similar study but take diet into account, since diet is such a major factor in the makeup of the gut microbiome. In conclusion, with the discovery of the role of HVA and the effects it can have on depression symptoms, as well as its connection to the gut microbiome and specific bacteria contained there, new avenues for treatment can be explored, and a small piece of this complex puzzle of depression has been added to help illuminate the big picture and will hopefully lead to the development of more effective therapies. Um, So here's some discussion questions that I provided. I'm not going to read through them. And then I also have my references after this. But I hope you enjoyed this paper. I really did. And I can't wait to hear what your thoughts are.